Hi, Assalamualaikum and good evening everyone. We are from Group 3 and we will present to you guys plant pathogen. Before we start our presentation, let me introduce myself first. My name is Nur Qurratu Aini binti Muhammad Mazuri 17181787/2. The contents for our presentation today are: the first one is introduction, the second one is plant pathogen, the third one is common pathogen infection methods, the fourth one is physiological plant disorder, the fifth one is epidemiology, and then the last one is disease resistance. Did you guys see the pretty patterns on the plant on the first slide that I show you guys? Well, it may look pretty, but actually it can cause harm on the plant. So, what is plant pathogen? Plant pathogen is an organism that causes disease on the plant. Disease on the plant can reduce yield and quality of harvested product. This is actually not very good as plant is our major source of food. Plant pathogen agent can be divided by two. The first one is abiotic and then the second one is biotic. Abiotic actually is a non-living things that can cause harm on the plant, which are sun, soil, temperature, atmospheric gases, and water. What about biotic? Biotic is a living things that cause harm on the plant. We have plants, animals, bacteria, fungi, and many more that will be discussed in this presentation. But we will emphasize more on biotic for this presentation. We have reached the second contents for our presentation, which is plant pathogens. We have fungi, fungus-like organism, bacteria, viruses, nematodes, protozoa and algae, and the last one is parasitic plants. Plant pathogen agent for fungi are ascomycetes and basidiomycetes. Ascomycetes is a spore shooters that produce microscopic spores inside sacs known as asci. The asexual reproduction in Ascomycota is responsible to spread fungi to new areas. In conclusion, we can say that this is how ascomycetes spread disease on plant. Basidiomycetes is a species of fungi that can cause plant disease on a plant called rust. Rust can affect many economically on important plants and usually appear as red, yellow, brown, rust, or even black powdery on leaf, young shoots, and fruits. So, how does basidiomycetes can cause disease on a plant? The plant growth and productivity will be reduced and some plants wither and die back. During their life cycles, rust pathogen will parasitize either one species of plant or two distinct species.
So hi, my name is Engku Nursyuhada, matrix number 1713936/2. So I'll continue with fungus-like organisms. So we have two here. The first one is omocytes and the second one is phytomyxia. So Omycota or omocytes form a distinct phylogenetic lineage of fungus-like eukaryotic microorganisms. They are filamentous and heterotrophic and can reproduce both sexually and asexually. So the phytomyxia are a class of parasites are cosmopolitans, obligate biotrophic protist parasites of plants, diatoms, omocytes, and brown algae. So next, omocytes. So they include some of the most destructive plant pathogens, including the genus Phytophthora, which includes the causal agents of potato leaf blight and sudden of death. Particular species of omocytes are responsible for root rot. Omocytes are capable of using effector protein to turn off a plant diseases in its infection process. So next we move on to phytomyxia. So some slime molds in phytomyxia cause important diseases including clubroot and cabbage and its relatives, and powdery scab in potatoes. These are caused by species of Plasmodiphora and Spongispora respectively. So here there's a picture of the phytomyxia, a plant caused by phytomyxia. So we move on to the third plant pathogen, which is bacteria. So most bacteria that are associated with plants are actually saprotropic and do not harm to the plant itself. However, a small number, around 100 known species, are able to cause disease. So bacterial diseases are much more prevalent in subtropical and tropical regions of the world. So most plants' pathogenic bacteria are rod-shaped bacilli. In order to be able to colonize the plant, they have specific pathogenicity factors. So we have 5 main types of bacterial pathogenicity factors are known. First, uses of cell wall degrading enzymes, toxin, effective proteins, phytohormones, and also exopolysaccharides. So significant bacterial plant pathogens, we have 3. The first one is Burkholderia, Proteobacteria, and the last one is Pseudomonas stringae, PV tomato. This causes tomato plants to produce less fruit, and it continues to adapt to the tomato by minimizing its recognition by the tomato immune system. So in bacteria, we have Phytoplasma and Spiroplasma. So Phyto and Spiroplasma are genera of bacteria that lack cell walls and are related to Mycoplasmas, which are human pathogens. Together, they are referred to as the molecules. They also tend to have smaller genomes than most other bacteria. They are normally transmitted by sap-sucking insects, being transferred into the plant phloem where it's produced.
Hello, my name is Idris Fahmi, and now we will be talking about viruses, and virus-like organisms. So there are many types of plant viruses, and some are asymptotic or latent. So under normal circumstances, plant viruses cause only loss of crop yield. Therefore, it is not economically viable to try to control them. Most plant viruses have small single-stranded RNA genomes. However, some plant viruses also have double-stranded RNA or single or double-stranded DNA genomes. These genomes may encode only 3 or 4 proteins, a replicase, a code protein, a movement protein, in order to allow cell-to-cell movement through plasmodesmata, and sometimes a protein that allows transmission by a vector. Plant viruses are generally transmitted from plant to plant by a vector, but mechanical and seed transmission also occur. Vector transmission is often by an insect, but some fungi, nematodes, and protozoa have been shown to be viral vectors. One example is mosaic disease of tobacco, where leaves are dwarfed and the chlorophyll of the leaves is destroyed. So, as an example, this is a tobacco mosaic virus. So we call it TMV. It does not have a distinct overwintering structure. Rather, it will overwinter in infected tobacco stalks and leaves in the soil on the surface of the contaminated seed. TMV can even survive in contaminated tobacco products for many years, so smokers can accidentally transmit it by touch, although not in the smoke itself. Tobacco mosaic virus is a positive-sense single-stranded RNA virus species in the genus Tobamovirus that infects a wide range of plants, especially tobacco. The infection causes characteristic patterns such as mosaic-like mottling and discoloration on the leaves. After its multiplication, it enters the neighboring cells through plasmodesmata. The infection spreads by direct contact to the neighboring cells, It can also spread through phloem for longer distance movement within the plant, and moreover, TMV can be transmitted from one plant to another by direct contact. Although TMV does not have defined transmission vectors, the virus can be easily transmitted from the infected host to the healthy plants by human handling. Nematodes are small multicellular worm-like animals. Many live freely in the soil, but there are some species that parasitize plant roots. They are a problem in tropical and subtropical regions of the world, where they may infect crops. Root knot nematodes have quite a large host range. They parasitize plant root systems and directly affect the uptake of water and nutrients needed for normal plant growth and reproduction. Nematodes are able to cause radical changes in root cells in order to facilitate their lifestyle. Plant parasitic nematodes are costly burdens of crop production. Ever present in nature, phytoparasitic nematodes are associated with nearly every important agricultural crop and represent a significant constraint on global food security. According to an estimate, plant parasitic nematodes are causing much more damage annually compared to insect pests. Root node nematodes, cyst nematodes, and lesion nematodes are ranked at the top of list of the most economically and scientifically important species due to their intricate relationship with the host plants, wide host range, and the level of damage ensued by infection. A crop yield loss due to these tiny unseen pests in various countries is enormous. Farmers or growers identify insect pests and other constraints as production problems, but often overlook plant parasitic nematodes. Nematode diseases are difficult to control because of their hidden nature and hence more often overlooked. Plant parasitic nematodes not only cause damage individually, but form disease complexes with other microorganisms and increase the crop loss. Also, the symptoms of nematode damage are not specific, resemble with the symptoms of other pathogens and abiotic stresses, such as water and mineral deficiency. There are a few examples of plant diseases caused by protozoa. They are transmitted as durable zoospores that may be able to survive in a resting state in the soil for many years. Further, they can transmit plant viruses. When the motile zoospores come into contact with a root hair, they produce a plasmodium which invades the roots. Parasitic plants such as broomrape, mistletoe, and dodder are included in the study of phytopathology. Dodder, for example, can be a conduit for the transmission of viruses or virus-like agents from a host plant to a plant that is not typically a host, or from an agent that is non-graft transmissible. Parasitic plants are among the most problematic pests of agricultural crops worldwide. They are found worldwide in all plant communities except aquatic. Parasitic plants are the organisms that settle in the host plant by means of the special organs they have developed and penetrate the vascular tissues of the host and meet their nutritional, water, and mineral needs from the host plant. This particular body they have is called a haustorium. The discovery and investigation of the haustorium structures led to the evolution of many heterotrophic plant species, previously defined as parasitic plants in different groups. Host organisms are very important in completing the life cycle of parasitic plants. In general, the parasite weakens the host, so it produces fewer flowers and viable seeds, or the value of the timber is reduced. However, some parasites, mostly annual root parasites belonging to the Orobanchaceae, can kill the host and cause significant economic damage while attacking monocultures in agriculture, and much effort is put into controlling these harmful parasites. Parasitic weeds are difficult to control because there are few resources for crop resistance and it is difficult to apply sufficiently selective control methods to kill weeds without physically and biochemically damaging the crop to which they are attached.
Hi, I'm Mohammed Hazwan. So I'm going to talk about common pathogenic infection methods. So there are four common pathogenic infection methods, which okay. So I start. One of them is cell wall degrading enzyme. So these are utilized to break down the plant cell wall so that the nutrient inside can be released. So Another one is toxin. This can either be either non-host specific, causing damage to all plants, or host specific, which cause damage to only the host plant. The third one is effector proteins. This effector protein can be secreted into the extracellular environment or directly into the host cell, with the three types secretion system being the most common route. So some of these effectors has been shown to suppress the host defense mechanism. This can involve things like lowering the plant internal signaling process and or lowering the production of photochemicals. So the last one is spores. This photogenic fungi can infect host plants by their spores. So the spores stick to the cuticular layer on the host plant leaf and stems initially. So the infectious spores must be transmitted from the pathogen source for this to happen. So they can be occur because of wind, water, or vectors like insects and people as humans. So when favorable condition exists, the spore will develop a germ tube, which is a modified hyphae. This germ tube then develops into an epithelium, which produces melanized cell walls to increase tube pressure. The epithelium exerts pressure against the cuticular layer in the form of hardened penetration pack once enough to tube pressure has been accumulated. So the secretion of cell wall disintegrating enzyme from the epithelium also aids this process. So the penetration pack forms a specialized hyphae called a haustorium once it enters the host tissue. So next is physiological plant disorders. So abiotic illness can be mistaken for pathogen-induced disorder in some cases. So natural processes such as drought, frost, snow, hailing, flooding, and inadequate drainage, nutrient insufficiency, mineral salt deposition such as sodium chloride and gypsum, windburn and storm breakage, and wildfire are all examples of abiotic causes. So humans or interference can also in can result in soil compaction, contamination of the air and soil, salinization caused by irrigation and road salting, over application of herbicide, clumsy handling such as land mower, damage to trees, vandalism, and uh, are, are included in the abiotic causes. Okay, next moving on to epidemiology. So what is epidemiology? It is a study of factors affecting the outbreak and spread of infectious disease. So elements involved with plant disease are best captured by a disease pyramid. The disease pyramid, which include aspects such as host, pathogen, and environment, serve as the foundation of the pyramid. Humans and time, in addition to these three elements, adds remaining element for form a disease pyramid. So in the past, there are epidemics that have caused massive losses in plants, such as Irish potato leaf blight, Dutch elm disease, and chestnut blight in North America. So the factors for the host, which is the plant resistant or susceptibility level, age, genetics, and the type of pathogen. So amount of inoculum, genetics, and type of reproduction. So in the center is the plant disease triangle. So next up is the disease resistant. Plant disease resistant refers to a plant ability to avoid and treat infection caused by plant pathogens. Cuticle layer, cell walls, and stomata gut cells are structures that assist plants to prevent illness. They serve as a barrier to keep infection out of the plants. So plant receptor begin signaling pathway to make chemicals that compete with the foreign compound once disease have crossed this barrier. Genes inside the host plant influence and trigger this pathway, which can be controlled through genetic breeding to develop plant kind that are resistant to damaging disease. And chemical deterrent of pest settlement and feeding, such as induction of defensive chemical, can be a key technique to lower herbivore damage towards the defense system. So that's all from us. Thank you.